Сабақ арасында фантамен үзіліс жаса. Эксперимент жасады, үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді, фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер, қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табыла ма? Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айнала ма? Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәлде болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм? И с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
The Kensington Kazakh Agritechnical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, KATU has trained more than 70,000 specialists, successfully working in all major sectors of the agrotechnical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan from the IAAR on the quality of educational programs. And also, KATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agro-Polytech University in Europe, and many others. Today, KATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. All necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an electronic system that has more than 1.5 million book collections.
One of the large multidisciplinary education institutions of Kazakhstan with a deep history is Awazov University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship academic mobility and double-degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At Awazov University, innovations are intensively introduced into the education system and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the laboratory of robotics and mechatronics at the laboratory of mathematical and 3D modeling, and provides material resources of the IT Center to students and teachers of the Awazov University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories and 2 sports complexes. For the comprehensive development of students, more than 30 public, political and cultural organizations function. Our university that has united history and modernity today prepares more than 27,000 students and invites to take your best.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жаса да үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді. Фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер. Қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табылама. Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айналама. Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәлде болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан, қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм? И с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
Zakensi Folin Kazakh Agritechnical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, KATU has trained more than 70,000 specialists, successfully working in all major sectors of the agrotechnical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan, from the IAAR on the quality of educational programs. And also, KATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agro-Paritech University in Europe, and many others. Today, KATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. All necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an electronic system that has more than 1.5 million book collections.
One of the large multidisciplinary education institutions of Kazakhstan with a deep history is Awezov University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship academic mobility and double degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At Awazov University, innovations are intensively introduced into the education system, and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the Laboratory of Robotics and Mechatronics at the Laboratory of Mathematical and 3D Modeling, and provides material resources of the IT Center to students and teachers of the Awazov University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories and 2 sports complexes. For the comprehensive development of students, more than 30 public, political and cultural organizations function. Awazov University, that has united history and modernity today, prepares more than 27,000 students and invites to take your best.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жаса да үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді. Фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер, қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табыла ма? Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айнала ма? Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәлде болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм? И с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
The Kensi Folin Kazakh Agritechnical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, KATU has trained more than 70,000 specialists, successfully working in all major sectors of the agrotechnical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan, from the IAAR on the quality of educational programs. And also, KATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agro-Polytech University in Europe, and many others. Today, KATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. all necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an electronic system that has more than 1.5 million book collections.
One of the large multidisciplinary education institutions of Kazakhstan with a deep history is WEINBERG University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship academic mobility and double degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At WEINBERG University, innovations are intensively introduced into the education system and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the laboratory of robotics and mechatronics at the laboratory of mathematical and 3D modeling, and provides material resources of the IT Center to students and teachers of the WEINBERG University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories and 2 sports complexes. For the comprehensive development of students, more than 30 public, political and cultural organizations function. WEINBERG University, that has united history and modernity today, prepares more than 27,000 students and invites to take your best.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жаса да үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді. Фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер, қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табыла ма? Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айнала ма? Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәлде болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан, қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм? И с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
The Kensington Kazakh Agritechnical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, KATU has trained more than 70,000 specialists, successfully working in all major sectors of the agrotechnical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan, from the IAAR on the quality of educational programs. And also, KATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agroparitech University in Europe, and many others. Today, KATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. All necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an electronic system that has more than 1.5 million book collections.
One of the large multidisciplinary education institutions of Kazakhstan with a deep history is Awezov University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship, academic mobility, and double degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At Awezov University, innovations are intensively introduced into the education system, and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the Laboratory of Robotics and Mechatronics at the Laboratory of Mathematical and 3D Modeling, and provides material resources of the IT Center to students and teachers of the Awezov University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national, and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories, and 2 sports complexes. For the comprehensive development of students, more than 30 public, political, and cultural organizations function. Awezov University, that has united history and modernity, today prepares more than 27,000 students and invites to take your best.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жаса да үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді. Фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер. Қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табыла ма? Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айнала ма? Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәл де болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов.
Society research professor. His research is in the area of oncogenesis and the genetic basis of human cancer. Robert Weinberg is a really awarded professor. He was given the Breakthrough Prize in Life Sciences in 2013, not to mention all the other awards he has received for his tireless effort to prevent people from getting cancer. And we would like to express our gratitude for joining Nobel Fest, the 5th Nobel Fest today.
In progress.
Yes. Good evening. Dobry вечер. Здравствуйте. Salam alaikum. Nice to meet everyone. I am here now. I'm not in Astana, but I'm in faraway Boston, and I am looking forward to describing to you how cancers arise within the human body. And here you can see in the first slide what things look like under the microscope as normal human colon lining in the colorectal tract begins to evolve progressively through multiple steps into high-grade carcinomas. When I say high-grade, I mean aggressive. Here below, we can see that this is a series of intermediate steps as there is the progressive evolution of normal tissue into hyperplastic tissue, into early benign growths, and ultimately a carcinoma, which is a primary tumor that may later on invade and metastasize to distant sites in the body. As it happens, one has discovered already more than 30 years ago that as cells evolve from normal to cancerous state, they acquire a whole series of mutations in their genomes, somatic mutations that occur, which propel these cells, drive them from a very simple state to a higher and higher grade of malignancy. And these somatic mutations, as they're called, have been studied through DNA sequencing and represent a compelling explanation of how primary tumors form. Having said that, I mentioned the fact that these multiple steps depicted here once again, but schematically, are repeated, are recapitulated in a whole series of other epithelial tissues throughout the body, where they create a whole series of common kinds of carcinomas, cancers that are common in many tissues and are seen in many countries throughout the world. And therefore, the principles I am describing today are not specific for colorectal cancer, cancer of the gut, of the colon and the rectum, but are typical for other kinds of commonly occurring carcinomas. Having said this, again, I emphasize that these mutations, which afflict these cells as they progressively accumulate more and more damage in their DNA, have been used to describe how primary tumors operate. But the provocative question is, how important are non-genetic programs, non-genetic factors, unlike these somatic mutations, which are surely genetic? How about non-genetic programs that govern the behavior of cancer cells as well? Are they important? Do they influence the behavior of cancer cells? Here, for example, is yet another depiction of what happens in pancreatic cancer. And once again, you see that as cancer cells evolve progressively from normalcy to high-grade malignancy, they acquire a whole series of somatic mutations that drives these cells to greater and greater degrees of abnormal proliferation. In fact, in the context of the colon, there are multiple alternative routes, genetic routes, roads that cancer cells can take en route from being fully normal to being capable of forming primary tumors. The first step is often affecting one of these alternative genes. The second step is affecting one or another of these genes, the third step and the fourth step. Together, they highlight the fact that cancer cells in human tissues can only arise after multiple genetic alterations, so-called somatic mutations, have been inflicted by the cancer cells. And one could demonstrate this, in fact, in my laboratory in 1999, where one introduced 5 different genes into normal human cells and converted them into cancer cells. These 5 different genes ostensibly altered 5 different regulatory circuits within the cells, and 5 were required. 4 was not enough. And I would mention in mice, in the cells of a mouse, only 2 changes are necessary in order for a normal cell to be converted into a tumor cell. So this scheme allows one to understand how primary tumors, in this case indicated as carcinomas, are able to form within various tissues throughout the human body. In fact, each of these changes, as indicated here on the right, through these experimentally introduced genes, is able to provide the cancer cell with a whole series of different behavioral abnormalities indicated here. These are the different hallmarks of cancer and indicate the aberrant or abnormal behavior of a cancer cell. So these genetic changes indicated here on the right collaborate with one another to create the cancerous state. Having said this, I'd like to focus on non-genetic programs, non-genetic processes that determine also cancer biology. And when I say that, I mean processes that operate without the intervention, without the involvement of somatic mutations. Here is one example of work that was done at Stanford University in California about 4 years ago, in which these authors used transgenic mice to inflict a common set of genetic alterations, somatic alterations, in 2 very closely related cell types within the lungs of mice. And these 2 cell types and the resulting tumors had the identical set of somatic mutations. But if one looked at the genes that were expressed in these 2 distinct tumors, arising in similar cell types in the body, but different, in one set of tumors, 978 genes were higher, and in another set of tumors, 1,138 genes were higher. And this emphasizes that even though these 2 sets of tumors had identically introduced somatic mutations, even though that happened, their gene expression patterns were very different. And this begins to indicate to us that one important determinant, one important governor of the biology of cancer cells already operates from the very beginning, which is to say, the biological program of the normal epithelium from which, in this case, the tumor arises. The differentiation program of the normal epithelium, which continues to influence all of the subsequent steps of cancer formation, multi-step cancer formation. To put it another way, it is not as if these somatic mutations that I cited before erase the influence, the pre-existing influence of the normal epithelial differentiation program. And this creates carcinomas that have various distinct behaviors depending on which tissue the tumors arise in. So this is one non-genetic or epigenetic influence, but there's yet another epigenetic influence, a non-genetic influence that does not involve somatic mutations that occurs here at this last step as primary tumors that arise in the area where the tumor first begin to spread throughout the body by invading and metastasizing, by spreading, by disseminating. These metastases are actually responsible for 90% of cancer-associated mortality. And here we can see that this single step, as I depicted before, is actually quite complex. It involves a series of sub-steps that are indicated here graphically and that are in aggregate together are termed the invasion metastasis cascade. You can see them here, and this is a rather complex series of steps as cancer cells from a primary tumor ultimately spread throughout the body, doing so by entering blood vessels, escaping from blood vessels, forming small colonies of cancer cells in distant tissues, and then growing to large colonies that form the macroscopic metastases that are responsible for the deaths of so many different cancer patients. Now, the complexity of this process provokes the question of how cancer cells can acquire all of the distinct capabilities, the abilities of these cells to execute successfully each one of these distinct steps that together lead to the formation of metastases. The fact is that additional mutations beyond those that were required previously to form the primary tumor, additional mutations are actually not necessary. This entire series of steps is acquired through a non-genetic mechanism, an epigenetic mechanism that is illustrated here. Here we see in this micrograph human breast cancer cells, which are expressing an epithelial protein called cytokeratin, and they're seen here in red. And these breast cancer cells are growing in a mouse host. The breast cancer cells have recruited, stromal cells have recruited cells from the mouse host, whose nuclei are shown here in blue. And what is of great interest to us here are these green cells, because these green cells derive from the red cells. The green cells have shut down the pre-existing epithelial marker, the red marker, cytokeratin, and now express instead a mesenchymal or mesenchymal marker termed vimentin. In other words, the red cells have shifted from the red state to the green state without any somatic mutations, ostensibly under the influence of signals that they receive from the nearby host cells, which are indicated here through their blue nuclei. And here you can see this more dramatically. Here is a tongue of carcinoma cells. Once again, they're expressing cytokeratin, which testifies to the fact that they are of epithelial origin. On the outside are the host cells that have been recruited into the tumor. Together, this is called the stroma. And what we see here is on the outer edge of this tongue of cancer cells, we see that these cells have shut down the cytokeratin epithelial marker. And here is another tongue of cancer cells. And the cells on the outside of the tongue have turned on the mesenchymal marker instead. In other words, these cells have moved from an epithelial to a mesenchymal state. And this change is often called the epithelial mesenchymal transition, or EMT. And obviously, it's occurring under the influence of signals that these carcinoma cells are receiving from the nearby stroma, from the nearby recruited mouse cells on the outside. Obviously, this is occurring without the involvement, without the intervention of somatic mutations. And here I can give you one example of this. This occurs at the edge of an island of carcinoma cells, indicated as before. And here we see that a cell from this cancer cell over here on the left, a carcinoma cell, releases one signal, in this case interleukin-1, that impinges and attaches to a cell from the stroma. The cell from the stroma may be a mesenchymal stem cell or a fibroblast, and the second cell responds reciprocally to this signal by sending signals back to the cancer cell, indicated here, 4 different signals that in turn induce the cancer cell to activate its previously silent, its previously latent EMT program, its epithelial mesenchymal transition program. So this program is totally occurring within the cancer cell under the influence of signals that the cancer cell receives from the recruited mouse stroma. And this program was first described and articulated by Elizabeth Hay already many, many years ago. And here you can see some indications of the complexity of this program as cells move from an epithelial state, indicated here, into a mesenchymal state. They become more mesenchymal cells rather than pre-existing epithelial cells. And here you can see some of the cell biological properties that cells acquire after they have activated the EMT program, a whole series of traits. In truth, there are as many as 400 genes that go up and go down as cells activate their EMT programs. So this is a very complex program that results in a profound change in the biology of a normal cell or a cancer cell, and that occurs without any additional somatic mutations. In fact, There are a whole series of different EMT programs. I indicate them all here very schematically. They share in common certain traits, but they may diverge from one another depending on the tissue in which the EMT program has become activated. Still, I will refer here to the single EMT program in what follows. We know that the EMT program is of ancient origins because if one looks in a whole variety of different metazoan embryos indicated here, one can find that in specific stages of embryonic development, there are different transcription factors. We call these EMTTFs, EMT-inducing transcription factors that are expressed locally and transiently in different stages of embryogenesis. You see there these very distinct expression patterns in a variety of different organisms and in a variety of different tissues. This indicates that the EMT program plays a critical role in normal embryogenesis and that these transcription factors, in fact, are the master regulators of this program. Here one can see, for example, that the expression of one of these transcription factors called twist is responsible for organizing the gastrulation in an early Drosophila embryo. The fact is that these transcription factors and the genes that encode them are present in the genomes of all metazoan species, indicating that they were already evolved 600 million years ago and have been retained with great fidelity in the genomes of all modern animals, all modern metazoa, including obviously ourselves. These genes that organize these different steps, you can see the names of these transcription factors, have been highly conserved, hardly altered in the last 60 million years. So these different EMTTFs, EMT-inducing transcription factors, act at different stages of embryogenesis, and they act in different combinations to orchestrate, to organize the different versions of the EMT program. Here is one manifestation of the EMT program, not in a normal developing embryo, but in the development of a mammary carcinoma within a mouse. Here we see that on the right, we can see the pattern of E-cadherin, which is a molecule that joins together adjacent epithelial cells, seen here in orange. And yet in this early stage carcinoma, there are cells seen here in this limited area where the E-cadherin, which ties together the epithelial cells, has been shut down. This is an important molecule in orchestrating and organizing the epithelial state. And in specifically this area, we see the activation, the turn on, the expression of one of these EMT-inducing transcription factors, which in this case happens to be called SNAIL. So here we begin to see proofs of the causality of these transcription factors in driving the shift from an epithelial to a mesenchymal state. Here's yet another aspect of tumor genesis that I'd like to move to, I'd like to shift gears to momentarily, and that is work that was undertaken by Muhammad Al-Hajj and Michael Clark already 18 years ago, in which they took a large number of breast cancer cells and they separated these cells into two subpopulations, with each of these dots here representing a distinct cell. They separated these cells using fluorescence-activated cell sorting on the basis of two cell surface markers that are expressed by cells in these two populations. One of these markers is CD44, and the other is CD24. And what they found is that there was a minority population, indicated here in this green square, and when cells of this minority population were implanted in a mouse, as few as 200 of these minority cells could seed the formation of a new tumor. In contrast, as many as 20,000 of the cells from the majority population failed to seed tumors in an immunocompromised mouse host. And this already indicated that these two populations of cells deriving from the same tumor and carrying ostensibly the same set of genetic alterations resided in two alternative states, one with high tumor initiating capability, the other with very low or no tumor initiating capability. Two alternative states. Those that have tumor-initiating capability, as demonstrated by their ability to seed new tumors, are often called cancer stem cells. And when one talks about these tumor-initiating or cancer stem cells, one imagines a hierarchy of organized epithelial cells, self-renewing stem cells, and more differentiated cells that operates within normal epithelial tissues. The cancer cells, the initiating cells, could be analogized. They're analogous to the self-renewing stem cells at the top of this hierarchy, whereas all the more differentiated cells represent the bulk of the cells in these tumors that have lost tumor-initiating ability. Here, I just show you another representation of the ability using fluorescence-activated cell sorting to fractionate these two populations of cells. The minority population of cells up here that has tumor-initiating or stem cell ability, the majority population, the non-stem cells, down here in the lower right, once again separated because of their display by the two cell types of these two cell surface proteins, antigens, called CD24 and CD44. Some time ago in my laboratory, Sender Rahmani and Wenjun Guo asked the following question. And why they asked this question, I still don't know, but they asked the following question. What happens if you put either the snail or the twist EMT-inducing transcription factor into these cells? Will that alter their behavior? It's not obvious that there should be any connection between the EMT program that I described before and the stem cell program that I describe now. What they found was, as might be anticipated if we look here at the lower row of images, these cells which previously looked very epithelial in monolayer culture, after acquiring either the snail or the twist EMT-inducing transcription factor, took on the shape of fibroblasts in monolayer culture in Petri dish. More importantly for our discussion, forcing these cells here to express EMT-inducing transcription factors drove the cells from the majority population all the way into the stem cell state, up indicated here in the top left. And this was the first indication that there's a tight interaction, a tight connection between the EMT program that we described before and the stem cell program that we describe now. Somehow they are interlinked in terms of their control mechanisms. This connection between the EMT program and the stem cell program operates not only in cancer cells, as I will show you later, but also in the normal mammary gland. Here is a cross-section of a normal mammary duct of a mouse, and the mammary duct is composed of epithelial cells that reside within two layers, the luminal layer on the inside of the duct and the basal layer on the outside of the duct. And as you can see here, it has long been known that the normal mammary stem cells reside in the basal layer, and these basal cells express the slug EMT-inducing transcription factor. Even though there is no tumor forming here, the expression of this slug is apparently needed in order to keep these cells in the stem cell state, preparing them for subsequent growth as the mammary gland increases in size. And here one can demonstrate that the expression of the EMT program is important for their stemness. One can do this by undertaking the following experiment. In a young female mouse, one can take the mammary fat pad, where the breast will eventually develop, and remove the initial mammary ductal cells over here to the left. This is sometimes therefore called a cleared mammary stromal fat pad. And into this cleared mammary stromal fat pad, one can then implant mammary epithelial cells. And if the mammary epithelial cells have stem cells in them, they will generate on their own over the next 6 weeks an entire normal mammary ductal tree, demonstrating their stemness. Here you can see what such an artificially generated mammary ductal tree looks like in comparison with a very similar one that is expressed normally. The question is, can the EMT program impart to the breast cancer cells, to the normal mammary epithelial cells, the ability to seed a normal mammary gland? If one takes 10,000 normal mammary epithelial cells and does not manipulate them further and implants them in a mammary stromal fat pad, nothing happens. But if one takes these cells and exposes them for a brief period of time to EMT-inducing transcription factors, shuts down the transcription factors, and then implants as few as 100 of those cells into a cleared mammary stromal fat pad, those cells, as a consequence of their having been induced transiently to activate their EMT program, can, as few as 100 of these cells, can now seed the formation of an entire mammary ductal tree in the mammary gland. So this suggests that simply by forcing these cells to transiently enter into an EMT-induced state, this increases by the factor of at least 100 the representation of normal mammary stem cells, and further develops the connection between the EMT program on the one hand and the stemness program on the other hand. And as Xinyi in my laboratory has demonstrated, this association can be demonstrated in a whole variety of epithelial tissues throughout the body, but not all of them. In double-layered epithelial tissues, but not in single-layered epithelial tissues. And how the stem cells in the single-layered epithelial tissues are organized is something we still do not understand. In fact, in the context of cancer, I will tell you there's a strong influence between the EMT program and the tumor initiating or cancer stem cell ability. Here, in this experiment, one looks at mammary breast cancer cells of a mouse, and if one has high expression of the snail EMT-inducing transcription factors, as few as 1 in 91 cells, when implanted into a mouse host, can seed the formation of a new tumor. However, in stark contrast, here we have a subset of cells in the mammary gland which do not express the snail EMT-inducing transcription factor, but express as an alternative the slug factor. And here, in these cells that lack snail, as many as 72,000 cells are required to be introduced into the mouse in order to generate a single new tumor. So here one sees a strong association between an EMT program and tumor initiating capability, the same way and in analogy with the association of an EMT program with normal mammary epithelial cells, as I showed you previously. Here is a demonstration that the EMT program can actually drive cells into higher grades of malignancy. In this experiment, human breast cancer cells that are very weakly aggressive were implanted in a mouse, and the resulting tumor was looked at 8 to 10 weeks later. And here is the tumor mass seen under the microscope. What one sees here is the outside border of this tumor mass, and to the left is the adjacent host tissue, which is not part of the cancer. What one sees here is that the outer limit of this tumor island is quite smooth and therefore suggestive of a lack of invasiveness, which is a prerequisite to forming metastases. Here, in contrast, cells were induced briefly to undergo an EMT through the actions of these introduced transcription factors. And under these conditions, one sees a highly invasive edge of the tumor where the carcinoma cells are clearly invading into a lipid-rich host cell stroma. So these cells have become highly invasive as a consequence of their having briefly experienced 6 weeks earlier an induced EMT program. More important, perhaps, is the ability of the cancer cells to spread, to disseminate from the primary tumor to distant tissues, in this case, to the lungs. So here is the ability of the cancer cells without any EMT to form either micrometastases, having dozens of cells, or macroscopic metastases, having thousands to millions of cells. Neither of these metastases are formed in the control tumors indicated over here. However, if one exposes for a brief period of time these cancer cells either to the slug or the SOX9 or this combination of EMT-inducing transcription factors and waits 8 or 10 weeks later, now the mice have as many as 400 micrometastases in their lungs, and 100 macroscopic metastases in their lungs. This experiment demonstrates on its own the causal role that the EMT program can play in driving cancer cells to express higher grades of malignancy. And in fact, if we go back to this diagram of the EMT process and the invasion metastasis cascade, we can now speculate that if one has a primary carcinoma and one turns on transiently an EMT program, as must happen in many primary carcinomas, the expression of this program is on its own sufficient to enable the cancer cells to invade locally and ultimately to go into blood vessels, to escape and seed micrometastases in distant tissues. In other words, the EMT program, a non-genetic program, an epigenetic program, is capable of conferring on primary carcinoma cells all of these steps, all of this occurring without the involvement of an additional somatic mutation. So the first part of cancer formation depends on somatic mutations, but these last stages do not depend on EMT, do not depend on somatic mutations. Importantly here, to my mind, even though the EMT program can allow cancer cells to physically travel from the primary tumor to distant tissues, to my mind, the last step of the invasion metastasis cascade, which we call here colonization, cannot be achieved by the EMT program, because when these cancer cells land in a distant tissue, they find themselves in an unfamiliar environment, and therefore they must somehow determine or figure out how they can assemble an adaptive program that enables them to thrive, to proliferate in a distant and unfamiliar territory. After all, why should breast cancer cells, which originated in the mammary gland, why should they thrive in the liver or the lungs or the brain, each of which presents a strange, unfamiliar, and inhospitable tissue microenvironment? And this last step of colonization, to my mind, is unlikely to be conferred by the EMT program, which instead is only able to physically move cells to distant tissue sites, whereas the last step must be a problem that is solved by the cancer cells after they reach these different distant tissues and begin to try to figure out an adaptive program that enables them, in turn, to colonize the tissue, that is to say, to grow from micrometastases, which may only have a very small number of cells, to macroscopic metastases. Fortunately, this last step is very inefficient, and therefore one can have women who have thousands, tens of thousands of micrometastases in their bone marrow, breast cancer patients, for example, and they will never form macroscopic metastases because of the inefficiency of this last step, which I propose is independent of the actions of the EMT program. So this remains the major problem of metastasis research. How do cancer cells figure out how to adapt and to thrive in a distant tissue microenvironment to which they are, it's unfamiliar, and to which they are poorly adapted at the moment of their arrival in that distant tissue? We know that different primary tumors have a tendency to form metastases to differing extents in target organs. So we know, for example, that the prostate cancer prefers to proliferate in the bone marrow. Colon cancer prefers to proliferate in the liver. Breast cancer, looking at the width of these arrows, prefers to proliferate either in the breast or the bone marrow, and so forth. And we don't really understand the reasons for all of these preferential metastasis formation behaviors, except to say that somehow breast cancer cells have a pre-existing ability to successfully adapt to growth in either the lung case, let's say the bone marrow. In the case of colon, solving this problem is not so difficult because, as many of you may know, the colon is drained by a portal vein which empties cells directly from the colon into the liver. So that must greatly influence and facilitate facilitate and enable the ability of colon cancer cells to form metastases in the liver. In any case, this is an area of intensive research at present. One could ask the question, if it's true that there are cancer stem cells in a primary tumor, where do they reside? How epithelial or mesenchymal are they? If you look at a micrograph of a typical tumor, there are cells in various stages, in various states of expressing either a mesenchymal or epithelial markers. It's a mixture of these different cells within a primary tumor. So that on its own does not shed any light, does not reveal where the stem cells reside. And so one can actually ask the question, how does one, if one pushes cells from an epithelial to mesenchymal state, do they initially go from E, epithelial, to M, mesenchymal, or is it a multi-step process? And here in this publication on single-cell RNA sequencing, one can see that cells that initially are in an epithelial state advance to increasing extents in a mesenchymal direction. And if one takes away, in this case, the inciting stimulatory signal, TGF-beta, they can return to via a mesenchymal epithelial transition to the epithelial state from which they initially emerged. Here's another way of thinking about the EMT program. Cells start out initially epithelial, and they don't simply switch to a mesenchymal state through the activation of the program. Instead, far more commonly, when an epithelial cell activates an EMT program, and this is true of both normal and neoplastic epithelial cells, they move first to one step, and then another step, and then a third step as they progress increasingly towards a highly mesenchymal state. So this is a multi-step process, and it is rare, as I'll emphasize momentarily, for cells to move from an E to an M state. This does not act like a binary switch, such as a light switch. Instead, it is a whole graded series of intermediate steps, and one can see that there are distinct intermediate steps here, as revealed by this single-cell RNA sequencing. So cells can enter into different states that have different combinations of epithelial and mesenchymal markers. And one can also illustrate this here. Here we're looking at a whole series of phenotypes where cells begin in the epithelial state, indicated here by E, and they progress to the mesenchymal state, indicated by M. And when a cell first activates the EMT program, it shuts down, as I mentioned before, as it moves into this program, it shuts down E-cadherin. It will then turn on later CD44, which is the cell surface markers I mentioned before. It will then turn on vimentin, another mesenchymal marker, and eventually it will shut off P63, a stem cell marker, and integrin beta 4. And when it goes even further, it will turn on fibronectin. So this is a complex series of changes, and this kind of depiction provokes the question of where the stem cells reside in this. And what one has found here in the work of Brian Beery is that the stem cells are not found over in the epithelial state or in the mesenchymal state, but halfway in between, in this quasi or half mesenchymal state. This is where the stem cells are, where they express a mixture of epithelial and mesenchymal markers. If the cells move too far in this direction, too far in the mesenchymal direction, then they will lose stemness, as indicated here. Importantly, I would mention the fact that in spontaneously arising tumors, cancer cells rarely, if ever, move all the way from an epithelial to mesenchymal state. That is, if anything, an experimental artifact. Instead, cancer cells activating an EMT program advance to differing extents along this axis, acquiring more mesenchymal markers and shutting down pre-existing epithelial markers. And so they are expressing a phenotypically mixed set of markers, both mesenchymal epithelial markers, as this program is activated. One should also mention, in passing, the fact that there are other consequences of activating an EMT program in cancer cells. I've just mentioned to you the fact that when cells activate an EMT program, they acquire the ability to renew themselves, which can be often depicted as self-renewal. So stemness is also another consequence of the activation of the EMT program. Importantly, cells which are initially chemosensitive and can be eliminated by chemotherapy become chemoresistant, and therefore the formation of of a more mesenchymal carcinoma cell confers on that cell resistance to different kinds of chemotherapy. Here one can see that one can kill epithelial cancer cells at relatively low doses of this chemotherapeutic drug here, increasing doses from left to right. If one looks at the viability of the cancer cells, relatively low doses can induce the epithelial carcinoma cells to die. But if they activate their EMT program, now one needs much higher doses to reduce their viability and to kill them. And that underscores the importance of this fact. This begins to reveal that the more mesenchymal carcinoma cells created by the EMT program are the enemy for several reasons. First of all, they're more capable of physically disseminating, that is to say, metastasizing. And secondly, they can resist elimination by treating the primary tumor with chemotherapy. And because of that, these resistant cells can survive, can persist, and ultimately, after the initial chemotherapy is stopped, these cells can then regenerate a new tumor, creating what is called therapeutic relapse, where the cancer reappears in the cancer patients. In this case, containing cells that are much more resistant to elimination by chemotherapy, by radiotherapy, and as I'll show you momentarily, even by immunotherapy. So these cells over here in the context of cancer, they are the enemy that must be in different ways eliminated in order to cure a patient who has a primary tumor. Here is one way of preferentially eliminating a cancer cell that has epithelial and mesenchymal markers. Up here we see in this graph, in this work of Whitney Henry, that if one looks at carcinoma cells that either have or have not activated the EMT program, if we plot on this graph the viability, the survival of cells, then epithelial cells at even relatively low concentrations of this drug, the epithelial cells lose much of their viability. They're almost all eliminated. Whereas the same cells that have activated an EMT, they are actually quite resistant to elimination, to killing by the EMT program, and they retain their viability, their ability to subsequently proliferate, echoing something I told you just before, that is to say that the cells that have activated an EMT program are actually chemoresistant. Here is one way, it turns out, of activating and eliminating the more mesenchymal carcinoma cells. One can activate a cell biological program in the carcinoma cells, which is called ferroptosis, and is an alternative to cell suicide known as apoptosis. This is an alternative mechanism of killing off the more mesenchymal cancer cells by treating them with this drug, which interrupts an enzyme that is responsible for detoxifying oxidized lipids in the cell. And what one sees here is the opposite behavior. If one uses this drug to shut down this enzyme, now the more mesenchymal carcinoma cells in the red line are killed preferentially compared with the more epithelial cells. So here's the exact opposite. And a therapy like this should be very useful in attacking preferentially the more mesenchymal carcinoma cells that are the subjects of so much pain and suffering in the case of a cancer patient. Here, I'm going to turn to another kind of therapy, not the induction of ferroptosis, which can be achieved preferentially in the more mesenchymal cells through the interruption of this biosynthetic pathway that ultimately results in cell suicide via the process of ferroptosis. I'm going to ask another question, which is the work of Anushka Dongre, who asked a different question. If we compare epithelial cells and mesenchymal cells, are they equally susceptible to elimination by checkpoint immunotherapy? And we know that checkpoint immunotherapy involves a series of controls which normally shuts down the ability of a T cell lymphocyte to attack a cancer cell. The lymphocyte above, the tumor cell, the target below, and this normally operative program limits the ability of the lymphocyte, the cytotoxic T lymphocyte, to attack and kill the cancer cell. And one can unleash this by, in fact, adding antibodies to a tumor, which can then shut down this checkpoint therapy. And this checkpoint therapy then enables the cytotoxic T lymphocytes to attack and eliminate more effectively the tumor cell. This is used very frequently in the case of different kinds of recently developed immunotherapies. And here we can see one major success. Using two antibodies, indicated here in the lower left, ipilimumab and pembrolizumab, which interrupt two of these aforementioned checkpoint controls. And if one uses these controls, these antibodies, what one can see in the case of metastatic melanoma is that patients who were otherwise doomed to die in large numbers, this is the proportion of patients who are still alive, if one treats them with these checkpoint blockades, a significant proportion of these patients are then treated in a way that causes the tumor to disappear and allows these patients to survive apparently indefinitely. To put it another way, this checkpoint immunotherapy seems to cure a significant proportion of melanoma patients, opening the door to the possibility that other kinds of tumors might in fact also be eliminated. However, in truth, to date, one still needs to develop these checkpoint immunotherapies to be equally effective in the case of carcinomas. And that raises the question of why many carcinomas, unlike melanomas, are resistant to elimination by checkpoint immunotherapy. Here, I'll show the work of Dongre, who used different kinds of mouse mammary carcinoma cell lines. These cell lines are stably either in the epithelial state or in the mesenchymal state. These paired cell lines, they have the identical set of somatic mutations. The epithelial cells reside stably in an epithelial state, as indicated by their expression of E-cadherin, but the absence of any expression of mesenchymal markers indicated here. Conversely, oppositely, the mesenchymal cells express a whole series of mesenchymal markers, but fail to express E-cadherin, which, as I argued before, is the epithelial marker. And she actually used two paired groups of cancer cells, one pair, E and M, a second pair of breast cancer cells, once again also stably either in the E or in the M state, either in the epithelial state or the mesenchymal state. And she asked the question, what is the immune microenvironment of these cells like? What kind of immune cells are present in the tumors formed by the epithelial cancer cells and the mesenchymal cancer cells? And here she looked for the presence of macrophages in the stroma of these tumors. And what she found was when she looked for immunosuppressive macrophages that are capable of shutting down an immune attack on the tumor, these immunosuppressive macrophages were present far higher in the mesenchymal tumors than they were in the epithelial tumors. And one can see this here, where in the mesenchymal tumors, these tumors had attracted and recruited immunosuppressive macrophages that shut down immune attack. Moreover, if one looks at the cytotoxic T cells with which the immune system normally attacks and eliminates cells, she found that these cytotoxic T cells were present in far lower numbers in the mesenchymal tumors rather than the epithelial tumors. And if one looked at the T cells that were present in the mesenchymal tumors, many of them displayed markers of exhaustion, which means these cells might be present in the tumors, but they are functionally exhausted and no longer capable of inducing any kind of killing of the cancer cells. Many of these exhausted T cells are present in the mesenchymal cells, but not in the epithelial cancer cells, one can also look for other immunosuppressive cells. For example, here's a subset of lymphocytes called regulatory T cells. And these immunosuppressive regulatory T cells are present in far higher numbers in the mesenchymal tumors than they are in the epithelial tumors. So far, I've shown you that the immune microenvironment of a mesenchymal tumor is much different than the immune microenvironment is of an epithelial tumor. And one can even find, for example, these immunosuppressive macrophages in the middle of a mesenchymal tumor where they shut down the immune response. But in the epithelial tumor, the immunosuppressive macrophages are exiled, are sent to the outer edge of the tumor, and are not permitted to enter into the tumor mass itself. And one can even show yet other differences between the immune behavior of the mesenchymal cells compared with the epithelial cells. For example, here one studies the presence of cell surface MHC molecules. And as many of you know, these MHC molecules are presented on the surface of cells and used by cells to provide a vision by the immune system of the intracellular proteins that these cells are making. The fact is that these MHC molecules are present in relatively low concentrations, if not being totally absent, in the more mesenchymal cells rather than the epithelial cells, meaning that the immune system cannot be aware, is blinded to the presence or the absence of certain antigens within the mesenchymal carcinoma cells, whereas in the epithelial cancer cells, the immune system can readily survey what is being expressed. And all of this begins to suggest that the epithelial cancer cells should be much more sensitive to elimination by checkpoint immunotherapy compared with the mesenchymal carcinoma cells. And here I give you some evidence of that. Here's an epithelial tumor growing, and what one sees here over a period of 30 days, the epithelial tumor is growing larger and larger and larger. But if one applies this checkpoint immunotherapy antibody, now this tumor is kept under pretty good control, seen here with this dashed control. If one looks at a mesenchymal tumor, as Anushka Dongre had done, working together with Mohammed Rashidian, she found that the mesenchymal tumor normally grows like this, and if she applied this anti-CTLA-4 checkpoint immunotherapy, nothing happened. The mesenchymal tumor didn't care at all whether or not a checkpoint immunotherapy was being applied. And this suggests that the state of the cancer cell, whether it resides in an epithelial state or a mesenchymal state, has a strong effect on the ability of checkpoint immunotherapy to eliminate the tumor, tumor cells. One interesting experiment was the following. Anushka Dongre took two populations of cells, more mesenchymal, more epithelial, and she mixed them together in different proportions. In the green experiment I'm showing you here, she mixed 9 parts of epithelial cells with 1 part of mesenchymal cells, and she made a mixed tumor. And she implanted the mixed tumor into mice and asked what the outcome would be after she applied checkpoint immunotherapy. The behavior of the epithelial cells, I showed you before, they're quite sensitive to the checkpoint immunotherapy. Here, the mesenchymal cells, as I also showed you before, are quite resistant. The mixed tumors, where only 10% of the cells in the tumor at the beginning and the end are mesenchymal, they're quite resistant to elimination by checkpoint immunotherapy, indicating that within a primary tumor, a minority of more mesenchymal carcinoma cells is sufficient to protect the epithelial majority of cells around them from elimination by checkpoint immunotherapy. And this may provide some explanation of why, in the context of clinical immunotherapy, most carcinoma cells are relatively resistant to elimination by checkpoint immunotherapy, if only because they contain minority populations of mesenchymal cells that serve to protect their more epithelial neighbors around them from elimination by checkpoint immunotherapy. And one can see such mixed tumors here. Here's an island of epithelial cells surrounded in a mixed tumor by these mesenchymal carcinoma cells. And importantly here, she asked the following question. If the mesenchymal cells are able to protect their epithelial neighbors, what do they use to do that? Do they release a whole series of secreted factors, which in fact are indicated here? And in her more recent work, as indicated in this table, she's found a whole series of factors that are released, signals that are released by the mesenchymal carcinoma cells and are able to protect the epithelial carcinoma cells from elimination by checkpoint immunotherapy. Here are these factors indicated one after another, and these proteins are synthesized and released by the mesenchymal carcinoma cells, and they can then thereafter protect the epithelial carcinoma cells nearby from elimination by checkpoint immunotherapy. And what she finds when she prevents the mesenchymal carcinoma cells from releasing one or another of these 7 factors, if she blocks the ability of the carcinoma cell to make this protein, now, once again, if you see here in the red epithelial, red lymphocytes are now, cytotoxic lymphocytes are now welcomed back into the tumor. The same with these 2 other factors, whereas previously in the mesenchymal tumor, there were no lymphocytes, cytotoxic T lymphocytes that were allowed in. Now, if one blocks the ability of the mesenchymal carcinoma cells to release either this or this or this, now cytotoxic T lymphocytes can move into the tumor mass, once again, in principle, showing their ability to kill the cancer cell. And here, just in a final experiment, I mentioned that we showed before that mesenchymal carcinoma cells, in this case expressing high levels of this EMT-inducing transcription factor, are fully resistant in the red line to elimination by checkpoint immunotherapy. However, if these mesenchymal carcinoma cells are prevented from producing this protein called CD73, now these otherwise resistant cells can actually be eliminated by checkpoint immunotherapy using the anti-CTLA-4 checkpoint immunotherapy. In other words, if one prevents these mesenchymal carcinoma cells from protecting themselves through the secretion of this CD73 molecule, now they become susceptible to elimination by checkpoint immunotherapy, which indicates the beginnings of an understanding of how one can convert the checkpoint immunotherapy into a mechanism, into a therapy that is useful in the clinic. And so here is a recent gathering of my group. Unusually fine-looking people, as you can see. This was last summer, deep in the woods of New Hampshire. And on that note, I would now be happy to receive questions, if any were posed. And again, I thank you for your attention.
Thank you so much. Thank you, Professor Weinberg, for sharing in such great detail this very important research. We are also thankful for all the scientists you mentioned and those who continue to contribute their time and energy towards cancer research and treatment. So there was some questions from the chat, just a couple. So the first one is the difference between equipment available to different universities, research centers, and even countries impacts the quality of research significantly. Are there any tips from your practice? How can we overcome the availability of research for those who are not at MIT, unfortunately?
Well, there are many places in the world where these experiments can be taken out, can be taken on, but in the end, there's a very simple answer to this question. It's one of money. In other words, gengi. One needs money to buy the equipment, to buy the microscopes, to buy the incubators to grow cells, to show immunofluorescence. One needs special microscopes. And so without support by the government or by foundations, it's simply impossible to conduct such experiments. I don't know what else to say, except that the world is a very unfair place. There is an unequal distribution of resources. And unless and until people in a variety of different countries, including those in Central Asia, are able to reach an economic level, they will be unable to undertake these experiments. not because they lack the brain power, but simply because they lack the money to buy all these pieces of equipment and reagents.
Yeah, that's true. You've been studying cancer for a long time. Have you noticed any patterns in the number of cancer patients? Maybe there are more people who are getting impacted by cancer. Maybe there are more children. Like, what's going on?
Well, in the end, it's clear that preventing cancer is much more effective than trying to cure existing cancers. And as a consequence, we now see, for example, in the West, smaller and smaller numbers of people dying from lung cancer. The fact is, this is directly attributable, this is directly caused by people stopping from cigarette smoking. It's not very mysterious. And so there's been a 50, 60, 70% decrease in lung cancer mortality, not because of the brilliance of the oncologists or the brilliance of the researchers. Instead, it's simply lifestyle factors have been changed. There has been progress in treating certain kinds of cancer. The mortality from breast cancer has been reduced by as much as 40% through a whole mixture of different treatments: chemotherapy, radiotherapy, monoclonal antibody therapy, tamoxifen, and so forth. And this is a major step forward. Unfortunately, however, many of the more common cancers that are highly aggressive, such as many kinds of lung cancer, pancreatic cancer, advanced colorectal cancer, are still expected to kill lots of people, and we still do not have good ways of treating such aggressive tumors. In the case of colon cancer, by the way, one can reduce colon cancer mortality by 60 to 70% by doing routine screening, that is to say, by doing colonoscopy, which can detect and eliminate early stage colon cancer tumors, thereby preventing that person from ever developing a life-threatening tumor. So there's a whole mixed bag. Some cancers are essentially incurable now, like, for example, pancreatic cancer, and have been for a very long time, whereas others are increasingly curable, such as breast cancer.
And the last very specific question, very close to your research, could you explore on how cancer metastatic cells avoid getting noticed by the immune cells during their migration to the new environment?
So the question is, when cancer cells leave the primary tumor and they go through the circulation, through the blood, they can be attacked. They can be ambushed by immune cells, notably natural killer cells, NK cells. And cancer cells are often able to protect themselves from being noticed by attracting and recruiting a whole cloud of platelets that attaches to the outer side of the cancer cells and prevents them from being attacked by the natural killer cells. Importantly, cancer cells spend only a very short time in the general circulation. Once they're released into the circulation, they might already be trapped in distant tissues 5 or 10 minutes later and begin to escape from the blood system into the parenchyma of the distant tissue. And so there's not a long time window of vulnerability, since the time of passage from the primary tumor to the distant tissue is actually quite short. Still, there is some loss of cancer cells in the circulation. The biggest obstacle to successful metastasis formation happens later, when the vast majority of disseminated cancer cells land in a distant tissue and fail for various reasons to form macroscopic metastases, in part, perhaps, because they're recognized and eliminated by immune cells.
Okay, thank you so much. And I think the last question, you mentioned that cancer stem cells keep mixed phenotype of epithelial state. Does it mean that you do not undergo AMT and thus do not metastasize migrate to keep their stemness?
Cancer cells activate a partial EMT program, which I showed you before, and that partial EMT program, without their becoming entirely mesenchymal, allows them to acquire stemness. And while they are in this partially epithelial, partially mesenchymal, mixed phenotypic state, they're able to escape the primary tumor and they're able to disseminate to distant tissues. Once they're there, once they're in the distant tissue, they no longer may need the stemness, which they would require to serve as the founders of the metastatic colony. And therefore, once cancer cells have disseminated, being powered by the EMT program, then they may actually inactivate and reverse the EMT program and undertake the reverse, which is sometimes called the mesenchymal epithelial transition, or MET, instead of EMT. And so in the distant tissues, they may then undergo this change, which puts them once again back into an epithelial state after they have disseminated, after they have spread.
Okay. So we have already passed our hour mark. So thank you so much for your time. Thank you so much for answering all the questions. We really hope to see you one day here in Kazakhstan, if the timing allows. So thank you for joining us from Boston, and we wish you a great day.
I thank you as well. Спасибо большое. Мне было очень приятно. And let's hope we will meet somewhere on this globe. Пока.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жаса да үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді. Фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер. Қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табыла ма? Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айнала ма? Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәл де болса жақсарту маңызды. Міне осындай адамдарға жусан керек. Жусан қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или